switch to something that is very interesting. Und ich gehe wieder in das Englische. Wir sprechen
the theory that there are no viruses, or if there are viruses, this particular SARS-CoV-2 virus has never been isolated in a scientifically correct manner. And we're going to talk to Dr. Andrew Kaufman and Dr. Stefan Lanka. Andrew Kaufman is an American forensic psychiatrist, natural healing consultant, inventor, and public speaker. And Dr. Stefan Lanka is a German biologist and author. I'm sorry to have kept you waiting for so long. But I hope you're still awake. Who's going to start? Yeah, great. Hi, Andy.
Hi, Reiner. Nice to see you.
Nice to see you.
Let me say hello to the rest of your committee as well, and good morning to Stefan, or good evening.
Sure.
If it's okay, I'd like to start.
Sure. Sure.
So I want to just thank the committee for inviting us and allowing this scientific narrative to be discussed on this platform, which is a very important platform.
Thank you.
I want to particularly thank Dr. Wodarg for his presentation earlier. And essentially, he gave the mainstream view of virology and immunology. And it's the same material that I learned in medical school, in textbooks, and by taking the published scientific papers at face value. So this is perfectly set us up for Stefan and I to demonstrate and explain how and why that entire narrative is false. And I want to clarify that we actually are not presenting a theory, because a theory would purport to explain a natural phenomenon. And what we are doing is actually refuting a theory, because we haven't made a claim about the cause of a disease, but there has been a claim made that there's a virus, SARS-CoV-2, and the claims have been made that there are many other viruses, that those viruses, one, exist, and two, cause a specific disease. And what we are doing is refuting the evidence put forth to satisfy that claim, because it is extremely faulty and perhaps even fraudulent at some level. So I want to make it just very clear that what we are arguing is that the methods put forth to demonstrate the existence of these so-called pathogenic viruses simply does not actually prove their existence at all. And there are two levels of research, or the way that I like to think about it. There's the primary research and the secondary research. The primary research are the experiments done to actually demonstrate the existence of the virus and its genetic and other properties. And that led to using the PCR procedure, which is really a manufacturing process to manufacture sequences of genetic material from very tiny amounts, of so-called amplification. And then the secondary research is all of the research built upon the presumption that there is this virus that causes disease. So this would include all of the clinical studies, the mortality studies, many other molecular studies based on variants and such. And almost all of this research is based upon defining the condition with the PCR test, as well as the primary research. So since we realize that the PCR test has never been validated even, that it's not really a test, that it can be manipulated in many ways, and its results are essentially meaningless, completely meaningless, we know that all of the research based upon that is all also meaningless, because it's based upon a false or meaningless premise. So I want to just also point out that many of the people giving the other opinion are not making scientific arguments about the existence of viruses. They're not talking about how the experiments actually show what they're purporting to show. Instead, they use logic fallacy. And a lot of that is ad hominem is the most common, of course, and Stefan and I have been called all kinds of names and criticized based on that. But also there are a lot of other false scientific arguments. Like, for example, many people say, well, look, there's 10 or more papers published that all say they isolated this virus. And of course, that is true. But the thing is, what we're saying is that they did the same experiment in all those papers, and the experiment is faulty. It doesn't actually show that the virus exists. So the proof that's put forth by the scientific and medical establishment, as well as the government and public health structures, that this virus exists is really based on three pillars. First, there are the so-called isolation studies. The second pillar are the electron micrograph images. And the third pillar is the genome sequencing. So I'd like to just refute those one at a time. In some very basic terms, I don't want to get into a lot of scientific detail here, but I'm happy to answer questions about any detail. I've read all of the papers and procedures related to these experiments. So for the isolation, I want to give you a little bit of a historical background to set the stage. And first of all, let me say that virologists had to essentially redefine the word isolation in order to conduct these experiments. And this is a point that's very, very confusing. So I'll try not to use that term in my explanation. But the origin of this goes back to the late 40s and the early 50s, when scientists in virology were largely concerned with manufacturing vaccines. And at the time, it was thought that in order to manufacture a vaccine, you had to use the type of tissue that the disease was in related to that virus. So, for example, for polio, you could have to use spinal cord tissue or nerve cells in order to manufacture the vaccine. But this was very, very difficult because those kind of cells are very difficult to culture in the laboratory. So along came John Franklin Enders with a solution to this problem. He tried an experiment using human embryonic fetal stem cells, or fetal cells, in a culture instead of the nerve tissue, and was able to manufacture what he said was the polio vaccine. And this made it much, much easier to do this manufacturing and to scale it up so that it could be distributed to the entire population. And as a result of these efforts, he was awarded the Nobel Prize. Now, he continued to use this experiment, which is a manufacturing procedure. So we have a parallel with the PCR, that we have a experimental procedure that was developed for manufacturing purposes that is converted into proving the existence of something. So he conducted the same type of experiments, but instead of using human fetal cells, he used monkey kidney cells, so-called vero cells. And this was published in 1954 with respect to the measles virus. And essentially, he followed the same procedure where he would take diseased tissue or fluid from someone with the illness and add it to a foreign cell culture in the laboratory, or tissue culture, and add additional ingredients to that, which have changed slightly over the time, but have essentially stayed the same. So, for example, in the measles experiment, he added milk to the sample of nasal fluid from the measles patients. We don't still use milk, but we use other sources of genetic material, such as bovine fetal calf serum. Antibiotics, which were used in that experiment, are still used, and depleted nutrition of the tissue cultures is also still used. And in that experiment, which was not purported to prove the existence of a virus, by the way, and you can look at the conclusions in that paper, it was shown that when they did the same experiment without a source of the measles virus at all, they also got the same results, which are the so-called cytopathic effects. And these are something you can see under a microscope that indicates in the tissue culture itself there is damage, as seen by a variety of different findings. And that just indicates the cells are in the dying process. And that is put forth after this paper by others as proof of the existence of a virus. And that experiment has essentially been carried forth since that time in the same almost exact way. and is still put forth to prove the existence of a virus in that clinical sample. But obviously, this is problematic. In order to show the existence of something in nature, you have to actually get it from nature, not from a laboratory experiment where there are other sources put in, such as cell cultures that are purchased, from a commercial facility, various chemicals and pharmaceuticals. So what they say, the reason that you have to do a cell culture, because before that, they actually tried to find virus particles directly from a biological sample, but they were never able to find a predominant particle that fit their theory of what viruses are. So when this experiment came along and was then purported to be the evidence, it was, kind of a heyday of virology because it was essentially a dead field with no proof of their concept. And now they found a surrogate marker to put forth as proof, and it seems that no one really scrutinized this properly. Now, so the reason they say they need to do a tissue culture is to produce enough virus to be able to see. However, the way that they say viruses live in a host that cause disease is the same exact thing as a tissue culture. So they would multiply, replicate in the host cells in the sick person, and then explode the cells and spread the deadly particles everywhere. And it should actually be the richest source of the viral particles because that is the natural place where the virus replicates. So it should be easy to take a sample, a tissue biopsy or a fluid, and find tons and tons of these particles and use physical means that have existed for more than half a century, such as centrifugation with a gradient density. There are more modern methods, such as affinity chromatography, which has been used to purify exosomes. So you can purify these particles directly out of a bodily fluid or a tissue. And further, there should be lots of microscopic evidence of direct damage from these particles, where you could see cells exploding from viral particles and viral particles infiltrating, everywhere in the images, but these images just simply don't exist. So because they're unable to show proof, they came up with this proxy experiment and offer it as proof. But now we know that the control experiment done by Enders in the 50s showed that it's the experiment itself that gives the results. And Dr. Lanka, and I'm sure he's going to talk about this, has repeated that control experiment in lung epithelial cells and showed the same results, that without any biological specimen at all, you get cytopathic effects demonstrating, the proof of a non-existent virus. Now, I want to just add one thing more about purifying the particles, because we know from admission of the authors of the publications, as published by Torsten Engelbrecht, through email correspondence, and we also have confirmed through Christine Massey's work on Freedom of Information Act requests, that no academic institution, government agency, or other organization anywhere in the world has purified virus particles. Now, Luc Montagnier, who is the Nobel Prize winner for the alleged discovery of the HIV virus, he said in an interview when asked, why is it important to purify virus particles, quote, to show they are real. Now I want to move on to the images from electron microscopes to point out how they also do not show proof of the existence of anything. And one fundamental aspect is that they come from these faulty tissue culture experiments. So they're not actually biopsies from individuals who are sick showing tissue destruction in the host, but they are essentially taken from these tissue culture experiments known as virus isolation experiments. Now, there's no way to identify what the particles are that are shown. But before I get to that, let me point out that many of the images that are put out there, even including by the National Institutes of Health on their Flickr account, where they put these images of SARS-CoV-2, an interesting place, many of these are simply renderings by an artist. They're computer-generated images. So I'm not talking about those images. I'm talking about the monochromatic electron micrograph images. Sorry.
It's ours.
Okay. So I'm talking about the monochromatic electron micrograph images. And one thing is you never see an image that has purified virus particles where that's the only thing present. You see a mixture of degrading cells, you see vesicles, you see them inside the cells, outside the cells, and budding at the cell surface. And this is a process of endocytosis and exocytosis that we read about. It's a basic biological process that you can see. And the particles that they're pointing at, there's no way to identify what those are. There's no specific label or stain that has been demonstrated and validated against an actual virus sample. So if you use, for example, a in situ hybridization with antibodies, those antibodies have never been demonstrated to be specific for any virus particle because there's never been a pure virus sample for which to test various antibodies for their chemical binding properties. We know from the HIV antibody test, for example, that over 60 different conditions give you have those same antibodies. So the specificity has to be clearly demonstrated through experiments. So what's happened is that we have published papers from the CDC and from the nephrology field where they found the same exact particles identical to what they call a coronavirus in size, morphology, and with the so-called spikes on it. But they found those before the COVID pandemic, and they found them in renal biopsies of people with various kidney diseases. None of the people had any viral illnesses, and this was obviously before it existed. They recognized that those proteins were actually not a spike protein in their sample. They identified it. I'm blanking on the name of the protein, but perhaps it will come to me later. Then we have another paper put forth, the rapid isolation and detection of SARS-CoV-2 from Australia, where they did the same tissue culture experiments and then took electron micrographs, but they didn't find any particles with the characteristic spikes. So what did they do? They took their sample and they added a digestive enzyme, trypsin, which digests proteins. Then they prepared another sample to look under the microscope and found the spikes. So in other words, they had a predetermined outcome and they did whatever procedure they could to make it look like the way they wanted it to look, and then they put this forth as evidence of something real that's from nature. So you can see that now we've shown that these images don't demonstrate the existence of a virus. And now we have the final pillar, which is the genome sequencing. And I want to distinguish this from how they sequence things like the human genome or the genome of a cat or a chimpanzee. Because in those instances, they take only cells from those animals and separate the nuclei and the DNA, and then they do various procedures to sequence that pure DNA from one single known sample. Now, with viruses, they have different mechanisms because, as I've stated before, they have never demonstrated actually having a pure virus in a test tube where they could directly extract the genetic sequences and genetic material from it. And then that would be valid if that were done, because that would show what it came from and that's what it was. But they're unable to do that, so they essentially use computer simulations as a way to model a theoretical virus without having any actual proof of it. And there are two techniques that are put forth. One is called, and there may be alternative names, but deep metagenomic sequencing. And the second one is called whole genome sequencing. Now, deep metagenomic sequencing is what is first used when there is suspicion of a new virus, previously unidentified and uncharacterized. And what they do is essentially take biological material, in most cases lung fluid, but in some cases nasopharyngeal swabs, from a individual that they say has a known illness. Now, of course, they've never done this on the same person where they've done a tissue culture isolation and then the genome sequencing from the same sample. And the genome sequencing originally was done from just one individual person who had a nonspecific respiratory illness and, of course, the requisite positive PCR test as the only proof that they actually had this non-existent viral illness. Now, what they did was take all of the RNA from that sample of lung fluid and separated out only the short strands because the technology that they use to actually do the sequencing only allows short strands as input. So that means that longer pieces, which could be like an intact gene or even a larger piece of the genome, which would at least tell you that everything in that one piece came from the same organism, they're subtracted out of the experiment. So only the short strands, generally 150 base pairs or fewer, are included. Some studies actually use shorter strands than that. And these strands are all amplified non-specifically using PCR, of course, which introduces additional error because every time you do a PCR amplification, there is an error rate because of mistakes of the enzyme as well as contaminants in the sample. And because of the power of amplification of PCR, something could be in there in, picomolar amounts or less and still show up in your result. So then once they amplify and make what's called a cDNA library, they sequence all of those short strands. And in the first case where they did this with SARS-CoV-2, it were over 56 million different sequences, and they call those reads. Now, that's the end of actually doing work with the physical sample, except for one small step at the end, because now it's all in the realm of the computer. So they put in those 56 million plus strands into not one, but two different computer programs, so they can guarantee that at least one of the programs will give them the result they want. Now, these computer programs take all the short reads, and they look for overlaps in the sequence, and they try to make them longer and put them together like puzzle pieces based on these overlaps. And these requires an immense amount of computation, and that's why a computer needs to be doing it. So what they did is they found that these two computer programs put together over 1 million different of these longer strands called contigs for contiguous strands. And out of those over 1 million strands, they picked the longest one arbitrarily and said, this is a template for a genome of a theoretical before unknown virus. And they ran it in a database, and they showed it was 89-point-something% similar to a bat SARS-like coronavirus genome, which was determined, by the way, by the same exact procedure. So we're now, at least 2 degrees separated from reality. And because it was 89% similar, they said it must also be a SARS-like coronavirus. Now, the human genome is 98% similar to chimpanzees. Yet we're a different, genus and species. So how could something less similar? And the similarity, by the way, is much more significant because the length of the genome, they say, for a virus is only 30,000 base pairs long. That's a tiny, way less than 1% of the size of the human genome. So if we have 98% similarity with chimpanzees, we have hundreds of millions or hundreds of billions of base pairs that are the same. If you have only less than 90% of a 30,000, base pair genome, it's a much lower percentage. And I want to also point out that the number of reads that were used out of the whole 56 million to create this one sample was 0.7%. So how does the computer know that only those 0.7% are with the virus, and how does it know the origin of any individual of those 56 million reads. We have no surrogate proof that there's actually a virus at all in this sample, because the only proof that's put forth is a PCR result, which we know only at best shows the presence of a short strand of RNA, right? And of course, we don't even need to do that, because we could show that same sequence in the contig sample if we wanted to. But there's no validation. This is, merely a simulation. There's no way to know what the origin of any individual of those reads was. There's no way to validate if the reads put into this contig are accurate or represent reality whatsoever. So this is simply a trick of computer simulation to put forth a theoretical genome of a theoretical organism that doesn't exist. So putting all of this together, we see that by using simple logic and reasoning, once we analyze the specific experimental procedures used to put forth the alleged evidence of the existence of this SARS-CoV-2 virus, are all highly flawed and defy logic, reason, and the scientific method. Therefore, the only rational conclusion is there is no virus shown to exist whatsoever. And of course, if there's no virus, then there's no possible way to test the causation of any illness. So we have to look for other explanations in order to understand what's going on.
Wolfgang, do you want to ask some questions?
Yes, I want to. Perhaps we don't have to we don't discuss the same thing twice. When Dr. Lanka tells his arguments, so I don't know how we organize this, because I have many questions and I think it's.
Maybe we should listen to what Stefan Lanka has to say first.
Would be good. I took my notes and then I have questions, and I think it's better when we do it like that, because otherwise we would discuss twice almost the same topics.
All right.
I agree.
So let's see. Let's hear Stefan Lanka in addition.
Stefan, you there?
Stefan Lanka, do you hear us?
It looks like he's dropped off. I don't see him in the participant list.
Oh my God.
Let me contact him on another channel and make sure he's okay.
Mm-hmm. He's back. Okay.
Ja, ich fliege hier dauernd raus.
Okay, great.
Okay.
I think you have to cut your the live stream.
I get kicked off all the time. Sie müssen den Livestream ausschalten.
Ich bin wieder dran.
Okay.
Yeah, that's right.
Now we can perfectly hear what you're saying.
Bin ich wieder online?
Ja.
Okay, wunderbar. Ja, ich möchte Andy danken, dass er in die ganzen Details reingegangen ist. Ja, ich kann zusammenfassen.
Summarize by saying that on all levels that we virologists do,
you have to close your browser. There is an echo. I closed everything.
then it should work now. Is it better now? Okay, I kept being kicked out and kept restarting, and I hope that it will work better now. Okay, thank you very much to Andy for summarizing all the details so brilliantly. If you take a closer look, we can see that the virologists, with their own tool, prove that they have disproved themselves. They do things that if you just take a logical look at them, that they create effects that they interpret as viral, generate a thought model at all levels, and claim that that is reality. Lay people can easily check that by seeing that all pictures that are to show viruses are only typical artifacts that occur in the test tube only, and there's no virologist who claims that they ever found it in any body fluid, nose, semen, blood, whatever. They're all specific artifacts that have no correspondence in reality. And with a view to SARS-CoV-2, now we can state that the virologists themselves describe step by step that in nasal swabs or other swabs, they can't find any nucleic acids that they can allocate to a virus. They have a first round of a very dirty PCR amplification. 14 cycles would be the right way. They go around 35 unspecific cycles and then find 10% of molecules that they combine to a sequence by computing methods. So they only get 10% of a sequence, then they add chemicals, the so-called primers. We know about them. There are the primers that the PCR needs. There are more or less specific ones, and they are look for sequences. These primers are synthesized, then they are put onto this mix that you've already amplified, and then you have enough starting material allowing you to calculate, to compute a so-called genome. That's only one of the contradictions that Andy has listed in so much detail now, namely that if you take a look at the finished construct, it's a mathematical construct, you can see the primers don't even match this. So what did we do? To prove our position, we took normal human tissue, treating it the way the virologists do, and didn't go through 35 cycles, and then again use a dirty PCR with 45 cycles, plus the primers, where the primers themselves are already 10 to 20% of the genome that you wind up with at the end of the day. Imagine this now. No, we have 14 clean cycles and we get the entire genome and can calculate it just the way the virologists do. So, in experimental terms, we have disproved the virus claim for SARS-CoV-2. I had done that already with the measles virus process in 2015 in a law case that I actually won in court by stating that none of the 6 publications contain any proof of the virus. I'm mentioning this because it's so important. One of the 6 publications is, and this is exactly where we lost the stream, I'm afraid.
Maybe he will be back in a minute.
Herr Lanka? Sind Sie noch
Mr.
da?
Lanka, are you still with us?
Andrew Kaufman, ist das wieder, oder ist das nochmal?
I think he got booted off again.
Ich glaube, er ist wieder rausgeflogen und der versucht jetzt wahrscheinlich gerade wieder sich einzuwählen.
Okay, warten wir noch einen Moment.
Wolfgang, haben Sie vielleicht eine direkte Frage?
Ja, ich würde gerne.
Discussion. I would try to separate between the big crime and the big fake, which we experienced since Wuhan, and the scientific basic arguments you have about the existence of viruses. These are two different things. You know that they are making us believe that we are to be afraid of some new virus is one thing, and I think we both agree that this is a very bad story, and it is not at all sufficiently documented. And there are two things: there are many very sick persons in a hospital in Wuhan, and who can die from some virus or even from side effects from drugs. We don't know. And then the second is that there is this laboratory where you can make gain of function things and experiment with molecules there. And then the third thing is the so-called Drosten test, which they used to define something, which was made in a way that they found as many so-called cases as possible. And we just heard from the Austrian attorney and from his client that they changed even changed the infection law in Germany and Austria to establish this term of cases. Before it was infections, it was illnesses. You can see an illness if some patient is ill, has symptoms, and this was used to identify infectious diseases. But now they formulated in the laws too. Some years before it started, they just changed those laws, and they spoke about cases, and this was used then. It was so easy. It's about defining. It's about new definitions, like the WHO defined the pandemic, and made a completely new definition. And they just use this power to define, to shape our image from what happens. And this is one thing, and it's a very bad story. And this is a long-running, the so-called pandemic preparedness thing opened the doors since more than 20 years, opened the doors for such tricks. This, I think we can agree. I think this is not the problem in our discussion. The problem starts when we speak about that all viruses are artifacts. This is what Stefan just said before, or the other thing, that there are no viruses. Oh, this is very problematic for me, and I don't agree at all, and we should discuss that.
Absolutely, Dr. Wardarg, and I think you make an excellent point about redefining, right? They changed the definition of a pandemic to fit their agenda. They changed the definition of a vaccine to include gene therapy to fit the agenda.
Herd immunity.
And they changed the definition of the word isolation.
Yes.
To fit their agenda of making vaccines. And so it's really important to look at this and to one realize that people being sick doesn't give you evidence of the cause of their illness. You have to do additional study, and I'm not sure why it's problematic. But if you have conclusive evidence, as I specified, that shows that one of these viruses exists, I would be happy to review it. And if it was an actual experiment that can demonstrate a pure virus particle taken from a natural source, like they show for proof of bacteriophages and exosomes and other particles on the nanoscale, I would certainly change my mind. But in my 2 years of research, reading tons and tons of virology papers, I've yet to come across one such experiment that shows the existence of a pathologic virus.
So you said that the virus that they about living or replicating viruses. This is already, I think, a failure in understanding, because viruses are not living. They are some package as information. They are they are produced by some cells, and then they are sent somewhere to some other cells. Sometimes, like the endosome, the exosome is sent from one cell to the other. The viruses are the same. They are packages with information, and they are sent from cells to other cells with some nucleic acid, and they give informations. They are a means of transportation of information.
Yeah.
And
so if that's not how we're told that viruses work. We're told that viruses cause cellular destruction and disease, not that they send useful information.
Yeah, but they don't replicate, but our cells or the cells of the host is producing them, is replicating them. And they don't do it themselves within the cell. They just give the possibility that the cell does it. Not all such packages that come into cells are permanently replicated. Sometimes they are put somewhere and they stay there. And you know that there is, it depends on how we understand the whole process, because what we observe,
whether the cells
replicate in a living
them through their process or whether the virus replicates itself, it doesn't matter, because the replication still occurs in the host, so you should be able to find the virus particles from the host. That's what I'm saying. I'm not, the model says that it uses the host machinery, but the viral genes actually code the new viral particles. So you could say it's, like a parasitic type of arrangement, or that you could say it's a symbiotic or it's a collaborative effort. But it doesn't really matter, because the important thing is that the replication of the virus is supposed to go on at the site of disease, and that's the cause of the disease. So that's where you have to find the viral particles to show that they're actually real.
How do you explain, all we call science and all we call knowledge is something which is always changed, and we learn and learn, and this is why we discuss such things. How can you explain that if you touch the saliva of a dog with rabies, that you may get rabies too, and you will die from it?
Well, I'll tell you, the answer to that is completely irrelevant, because I'm talking about demonstrating that there's a virus in that saliva. Why not purify viral particles from that saliva, show them to exist, then we can have that discussion. There could be many elements in saliva that could transmit disease, such as toxins. The reason why someone with rabies is salivating is to remove unwanted things from the body. That's why we have diarrhea, that's why we have coughing, that's why we have a runny nose. And it's no different in rabies, but the delivery transmission is much different in rabies because it's delivered by injection. It requires a bite. If you drink the saliva of a rabid animal, you will not get sick, but if it's injected and bypasses the normal... Okay, well, so let's say that it is true. It still doesn't matter, because you have to demonstrate that you can isolate the rabies virus from the saliva. Is that been shown? Do you have a paper showing that?
Don't ask me for such papers now, but if you...
Well, this is logic fallacy. You're not arguing the actual point about the proof of a virus. You're giving a tangential evidence that doesn't demonstrate the presence of a virus.
I don't speak about bad laboratory demonstrations or bad methodological mistakes. I speak about an explanation. How can some set of symptoms that are typical, like the smallpox, for instance, how can they come from one child to the next child in a very timely order? With direct, you can see the contact, and you can see the smallpox afterwards in the next child. You can see the bubbles which open
How do women synchronize their menstrual periods? Is it because they pass a virus back and forth? There are other ways that we communicate with each other besides passing viruses. In order to say that it's a viral mechanism, you have to show the actual virus. You can't just say, look, it passes from person to person. It must be a virus. Because you could say it must be an alien that causes the disease, and they pass an alien from person to person. It's the same argument. the three pillars are the only thing that are put forth to prove the existence of a virus. If you can explain how those pillars have been proven, or how I'm wrong, then you can make an argument. But saying that we don't have another explanation at hand is not an argument. That's the logic fallacy.
You're just coming and telling me all the experiments don't show anything, but you don't have a better explanation for transmittable diseases, which are said to be viral.
Well, I do, but it doesn't matter what my other explanation is, because what I've shown is that the explanation is not a virus. And then you have to do additional research to show what the real explanation is. And because germ theory has been the dominant paradigm in all of the scientific research in the 20th century, there aren't researchers looking for other causes, because they all just presume it's been caused by a virus.
There are many toxicology people who try to find out the effects of toxic substances. And I was working in this field too. It was always very difficult to find out what is the reason. And we know all those agents or viruses that are in the water, for instance. In summer, you have all those gastrointestinal.
So there are no virus particles purified from the water either.
Interrupting me.
Yeah, could you give Wolfgang the time to finish his thoughts, and then you could.
Yeah, I apologize, but he's not actually arguing what the points that I made. He's making tangential points that don't really speak to the existence of viruses. So you could see that it's kind of frustrating for me to just sit quietly and hear irrelevant evidence. You know, I can't object like in a courtroom and say irrelevant, but that's what you would do probably in the situation.
I think we should really just for instance, the example that you gave with this menstruation thing, I don't think that's a comparable thing, because there might be like some hormonal or other things going on, between the, that's
so
that's
couldn't
not
there
comparable.
be a hormonal or other thing going on when people get colds too?
Who knows? But is that your explanation?
Yeah, exactly. Exactly.
Excuse
Who
me.
knows?
Excuse me.
It's not relevant to whether viruses exist.
Sorry, could I finish my sentence also? Because, I think I'm not a doctor, so I'm just picking up your example, and I don't think that's matching the virus idea either. So, but I think let's just, because we want to have a discussion, I think it's not about throwing out ideas in a courtroom or saying this is not relevant. I think it's about having an open discussion, and we don't want to, it should be open. And now I think Wolfgang should finish his thoughts.
Yes, I listened to you all the time, a long time, and I didn't interrupt you. And I was not content with many things you said, and I need some time to try to mention some of it, at least. And I just spoke about the fake of this, what happened with the so-called corona, with so-called SARS-CoV-2 virus. And on the other hand, I speak about the clinical experience, and as a doctor for hygiene, all the reasons for successful and for unsuccessful preventing diseases, and about the differentiating toxicologic effects from infectious infects. This has a long story, and you have to learn a little bit more than this, what you cannot learn in laboratories. And so, if I'm not a laboratory guy. I do not do such research myself. And this is, what interests me is whether you have better explanations. As a scientist, you have to come and you have to say, I know it better. I can give you another hypothesis for something. You cannot just say, I'm not interested in that people get ill. I'm not interested when they touch the saliva of a dog. I'm not interested. The others, with the virus theory, they invented an injection which protects against infection with rabies, which was successful. People don't die when they get in touch with the rabies dog after they are vaccinated. And so this you can show this. And this is. Oh, das ist schön, wenn ich denn Deutsch sprechen kann. Wunderbar. Das ist mir viel lieber.
Ich falle dauernd raus. Ich fahre halt immer den Computer wieder hoch. Jetzt arbeite ich mit der Kamera.
I'm working with the camera in the computer. Somebody's got their browser open still.
The browser is closed. I close it. Is it better now?
I think so. Okay. Mr. Woodrock, I think it's a pity. In May, we had a live meeting, a Zoom meeting with a couple of other people, and you said we are now clarifying that question, and that hasn't been happening so far, and I think it's a pity that we are wasting time with something that we could have done at the time already, or if you had read my book, my documents, you could have understood what's going on.
Well, that's exactly what we want to discuss. So my understanding, Mr. Lanka, is that Andy says, where is there a true photo of a virus? And the answer to that is still outstanding, and we're still trying, or not me, we are, Mr. Wodak is trying to deduce it from other circumstances, and Andy feels that this is not good enough. I can understand that, but now this takes us a step further. Now it's your turn.
All photos that are to show photos are from the in vitro tube. So they are in the saliva, in the blood, somewhere elsewhere. So it's typical artifact that are only found in vitro in the test tube and no elsewhere. As a young student of biology, I went into this because I stumbled upon a structure which I thought that would be a harmless virus. It looked like something that we see as a virus today, and the finding is that these are minimum phaguses, spores, and these actually exist. But I can take all of these are not on bacteria. I can put all isolated spores and phaguses on the bacteria, and nothing will happen to the bacteria. But this is going to be used as a new model. And then these structures, in the case of the phagus, that are quite clear, every student knows these, isolates them as a whole, photographs them as a whole, and from every insulation, all viruses, all proteins assigned to that virus, and a nucleic acid, always of the same length, always of the same base pairs, the sequence. And this is something that led the virologists to believe that this is what viruses have to look like. But they didn't learn this as something that can be read out in a book by Professor Luck from 1990 on the early history of virology. And this overview work shows us that virologists did tests and proof research from healthy tissue that were then interpreted as virus strings, and that finished the theory. And they looked at all the transfer experiments, and it is how they put them in the brain or in the eye or in the lung, that it was exactly these expressions that create the symptoms that are then presented as transfer. And that is the case as of today. Since then, no transfer experiments have been published where there were any any control experiments. And this hasn't happened. And this is actually state of the art that I have to counter-check all of these things. So science, a science area that did without control experiments is simply not scientific, and none of the statements could be used. So as I'm here doing the split, believing in HIV and having found a structure that I think is a virus. And from that, I got the honors of an Austrian professor running the biggest sequencing company, GAC, building up the sequencing technologies. I grew up with this from 84 onwards, and he asked me whether I checked it with HIV. And I said, no, the whole world says it. And he says, no, I didn't ask the world, I asked you whether you checked it. And that embarrassed me. And so we started to discuss this. And this is how I went in here, step by step. HIV were to be very different viruses, a virus that is lethal. And then later it was found out that Gallo did manipulate it with hydrochloric acid, and the French who got the Nobel Prize said, my virus, if it holds true, changes the white blood plates. And as that was made public, that Gallo did the manipulation to build a lethal virus, well, Montague, we said, your virus only replicates then, and if they would stop to inhale the fungi, they would stop. And if you take clean water and vitamins, they would get the virus out of their body. That's what he hated all the time. But he gets the Nobel Prize for this in 2008. So I thought, if that's in the news, that Gallo manipulated to build a dead virus, then politics knows this, and this is why politics would use that in order to create fear in the population. So many of the people that I have seen in the presentations killed themselves because they didn't want to wait for the virus to kill them in 2 years. And that was quite hard for me to stand this emotionally. I couldn't help. I couldn't do anything. And at the same time, I was in the trap of fraud, of seeing this crime. And that damaged my work in clarifying these things. And later on, I tried to look for the origin of these concepts, and I found them in history that many people describe. Chermos O'Mahony, showing that medicine is completely bonkers. And this was someone who predicted Corona in 1976. If we continue with the medicine the way it is.
Let me not interrupt you, but we're under pressure of time now, and this is really interesting, and we all believe, of course.
I can talk about the vaccination consultation center that I run for a couple of years. We would need much more time for that.
Yeah, that'll take us astray now. We have to focus a bit, maybe find a point.
I think the most important question or the challenge that we have is the majority of the people believe in a terror biology that our own molecules form a terror group that wants to kill us, and that even travels. That's the idea. And the dying of people with this diagnosis is the proof that this evil must be around. And if we have belief in metastasis, they believe in flying metastasis, and then they are annoyed by us, by me and you, who say wearing masks doesn't help. For them, we are the terrorists because we come up with this concept. We question the concept. And if I had the great luck, I am working on this scanning technology. There's a whole area of knowledge which one could call psychosomatic, which explains these things, why people get sick one after the other or at the same time without gaps and no contradictions. And to popularize this, I am building a brain scanner with a technology where you can apply that to the brain in order to popularize this knowledge and take the fear out of this system, because finally we are not facing a virus of power. Of course, power uses fear, but we have a fundamental fear here covering all the population, and Corona is a massive awakening call because the fear is detached. I have written about this. I'd be happy to report in detail on that. And I'd like to ask you to put the link of Andrew's film, Terrain, explaining all this at top level, what other explanations there are, a better explanation for health and life, development and sickness and disease. So I hope that we have the opportunity here now to look at this, and in all peace and quiet, which is necessary for this topic, that we can find that. Of course, there's lots of things to say. I'm very sure of what I'm saying. I've taken through all the structures, the points. The structure I discovered is viroplankton today, and there is a structure which actually exists that may serve communication of nucleic acid, but there are other things, fantastic knowledge, and just one author, to just name him, is Bruce Charlton. He has been saying a long time, science is dead. Erwin Charkov could be my teacher, written in 1978. He predicted how all science is degenerated, and somebody who brought it to the point in 1929 already was Eugen Rosenstock-Huessy. I think he's the strongest thinker that we have. Unfortunately, he went elsewhere in 1973. The crossfire of reality, where he describes why the material explanation of life prevents us to take note of developments and find out something new. And he describes another aspect, which is if a scientist is paid by government bodies, of course he won't like to be contradicted by newer findings, and this is why they will automatically do pseudo-research in order to get away from truth. And then I find this, since 1929, it's unused. They are researching on cancer after old rules. And that's a double critique of the infection theory that have been developed from that point. And I just wish that you and the audience can read this up. I have my channel for that. Great people did it on Telegram, which is being filmed, Lanka Vision. You can find that and these are links to in-depth information. Project Emmanuel, Mr. Wodak, would be very good. Project Emmanuel, the German philosopher.
Well, in scientific discussion, it's always important to disclose your secondary interests. I think it's good that you said that you are working on this device and are invested there and have a business interest in that. There's a lot of discussion. Can you please let me finish? I can only say of myself that I don't have any secondary interest. The only thing that I've done were my publications, and I don't get any income from this. I only have damage. I'm hurt myself by what I do. Apart from all the good human vibes that I get. But I'm a bit annoyed about the level that we've been discussing here so far, because we would have to go into more detail. You have to prove to me that there are no viruses. Can you please let me finish now? You would have to prove, you would have to clearly prove and publish it, and it would have to be scrutinized. And it's not enough to make a TV broadcast or to go public on a show and claim this. What you achieve here is to distract people from the alternatives they have. You try to attract attention to your theory, distracting from other questions that need to be answered urgently, and you use what we have in common, namely that we're both against this Wuhan fake, but you superimpose your own interests, and I'm really annoyed by that.
Well, just look at the mirror and criticize yourself. I have published all of it. It's all online. All the texts are there. They are free to read.
But not scientifically published.
Whether it's scientific, Mr. Wodak, it's the peer review stops that new things are published.
That's too simple of an explanation.
We are just publishing the control experiments with Dr. Kaufman and Andy Kaufman because no scientific journal accepted it. They don't like the change of what they do. And that's the deepest argument that I've heard. The arguments, logics, naming facts are not appreciated because they don't have the stamp of the governmental knowledge. That's really wrong.
Well, Mr. Lanka, can I say one thing, please? I think we should use proper language here, and I don't think that it's appropriate that somebody else says something that he is way off the mark, and I don't want to have this sort of thing, this disrespectful language. I think we got together here. May I, Mr. Lanka, may I briefly say something? Mr. Lanka, can I briefly say something? We've been on the phone together before, and we discussed this very at length, and we met personally, and I thought that I can't say anything on the scientific merits, but that we could have an interested exchange of ideas here. And I don't want to have something here on the committee like we never had, that we laugh about what anybody says, or that we do not treat ourselves respectfully, treat each other respectively. And we're all interested in stopping these things, and I know that you're interested in that as well. And we've discussed multiple times here that there are many things in medicine that need to be rethought. Maybe you have interesting approaches here. I can't judge it. I'd say a lot of the experience that Mr. Wodak mentioned and many others are relevant too. And I think we can only speak at reasonable, in a reasonable way if we are respectful with each other. I think that's very important.
Well, you just can't. The killer argument, as in that arguments have no value because the pseudoscience doesn't publish it. I think, well, you have to get the information.
For 13
this
years.
This is a book written in 2019.
Can you just, Mr. Lanka,
Well, this argument is something that I don't see, and personally attack me and say I follow my own interests.
Mr. Lanka, can we please stick to the topic, and can we really focus on this viral issue if we want to continue this conversation? But I don't think it makes sense to go into you said this, now I said that, and let's simply step back. We don't have that much time anymore, but let's try to be productive talking about the topic. And I find, of course, that it's interesting that the idea is that we can talk about psychosomatic conditions that and that might transmit chickenpox. So that would be interesting. It's a new way of thinking about this. We would have to think about whether it's possible. There might be other possible explanations as well. And maybe the virus is that we always thought were the cause. I can't say anything about this. I've always thought that I don't get the flu because I'm psychosomatically infected by someone else, because there needs to be some transmission mechanism. We would probably have to have a more extensive discussion on this, but I think that should be in a different emotional setup. We are not fighting each other here. If anything, we would like to have an exchange of ideas. That makes sense.
That was my initiative and my idea, because people ask me what this theory is, that there's no viruses, and I said, well, I can't understand this, and I was open to invite you here, and I said, well, I would like to clarify this and talk about this topic, and I didn't get that far yet.
Maybe we simply should recognize that there are two different approaches here, but that in many areas there's some common ground as well. We're all convinced, after all, we've heard so far, that first of all, this is a non-existent pandemic, that it is a staged pandemic, long planned. Secondly, that they have a different agenda that has nothing to do with well-being or the health of people. And thirdly, that what the upshot is that we can see, i.e. crimes, serious crimes, that they need to be prosecuted in law. And that is what this is all about. We are convinced that the population as a whole has to rise, but the cleanup work in order for the population to be able to take action. And once we have done the cleanup, then the upshot has to lead to criminal investigation. There must be compensatory payments and long prison terms. So at some stage, we are already in lawsuits involved. We're in court. Sooner or later, we will be there. And then the question arises, what can we prove? What cannot we prove? So in the question of are there any viruses or not, and if we find that there are viruses, has this one been isolated? I don't think we'll reach agreement tonight in this aspect. But in terms of the preliminaries, we are on the same wavelength. We're also on the same wavelength when it comes to... We're also on a wavelength in terms of the economization of health, that the pharmaceutical industry is not a pharmaceutical industry, but really a pathogenic industry, because they can only earn money if we are all sick. We're on the same wavelength here. And if we arrive in the courts, and we are already there, and will continue to do that, then we are agreed, and I think we all can agree that, then we won't make progress if we debate in court whether there are viruses or not, or whether this one has been isolated or not. And again, we have common ground here, that this PCR test, no matter how it's applied, it is completely impossible to determine an infection. We've heard enough about the details and repeated them enough. And secondly, we also agreed, as that's my understanding of what you said, that these PCR tests by Drosten and the CDC test, produce masses of false positives, had to produce them because they wanted this, so that you have cases that are completely irrelevant because they are not based on any diseases, on any infections. I think that we can agree on that, or don't you agree? Can we hear the answer now?
Well, I'm convinced. I've been doing this for 30 years. If we don't learn from this, we are going to repeat it all. The virus is the crucial point. And if you present to a court the the non-scientific approach of the virologists, showing by the complete lack of control experiments, and come to the logic conclusion from the publication to create the Wuhan genome. There's only one, and it's been copied by everybody else. That virality itself has been disproven. Then you'll have the only effective legal means to say the Infection Protection Act requires a scientific approach, and that is not given.
Well, Mr. Lanka, but I always try to go the safest road. As a legal expert, I always try the safest approach. And the safest approach is not to engage in a debate that may be irrelevant at the end of the day, because I may achieve my goal faster some other way. If I start debating in court whether there are viruses or not, 99% of scientists claim that we have them, then I have a debate that costs me a lot of time. That doesn't take me to the objective, and the target must be to prove that it's a staged pandemic, and that is best achieved with the PCR test. Because if I go to court and I prove that the PCR test can never prove any infection, and secondly, the way it is done here definitely produces false positives, and we have the environment as well. The World Health Organization will prove that they were deliberately provoked because they wanted to have false positives. They wanted to have the international situation of public concern. That takes me to the target faster, because if we have 99 experts that the counter side will say that there are viruses, and 3 experts say there aren't any, then I'll use the case and a lot of time.
No virus is going to swear that they did the control experiment.
Mr. Wodak has an important point as well.
I'm completely convinced of that. You go your way, I go my way, and I'm sure that I'll deliver our common goal before you. We'll see that.
Well, we'd be all happy if you did. The more shots that are fired...
Maybe we could listen to Dr. Wodak, what he has to say.
Well, it's also a question of definition. If there are viruses or not, then it's a question of definition. I could also start a discussion about whether there is or isn't a pandemic. So there are no diseases that spread globally. That's a question of definition. The WHO has shown us how you can simply change everything by changing a definition, and then the reality changes. I could claim there's no herd immunity, or immunity is something that doesn't exist. I can claim all these things. And then you always have a discussion about this, just like if somebody claims that there are no viruses, it just won't help us, because with these theories that you hope to prove, they have to be so relevant that you can act in a relevant way with a reasonable result. And if you can't give me a reasonable result, how I can protect people from rabies or other infectious diseases, I have to say, Mr. Lanka, that's not good enough.
Well, you don't read my documents and said I didn't deliver. Read my papers. You'll find the answers to all your questions. That's not my knowledge.
I read a bit of it.
It's been collected over the years, and quite clearly, I was under pressure. I knew I couldn't explain these diseases, and maybe that's my major work, which I've done for many years. When I asked people, I was asked, should I vaccinate or not? I told the I told the people, ask the authorities for a scientific proof. And
Have you had patience?
And since 2000, year 2000, I couldn't answer this question. I didn't do this split to follow some kind of theory that I wasn't really convinced of.
But getting back to reality, patients who depend on help, and there are tried and tested methods that are helpful, and there are methods that make them even sicker. Unfortunately, we see a lot of those. And then there's wrong approaches. And that's where you have to help each other to find the right approach. And that is the function of science, or science in medicine, really. And I'm not necessarily used to, oh, I know better. I'm used to a questioning approach. That's why I invited you, and that's why I thought it was important. I have to say that this discussion, I find it disappointing. And if we start with these discussions about definitions, then we have to start from scratch with many things. These infections, there are constructed infections of cell cultures, or the virus proliferation happens under certain gelatin layers, etc. But that's the normal method, and I was told, I learned from people who do it in practice, virus purification doesn't leave the lab at all.
Virenpurifikation funktioniert gar nicht so, sondern sie werden unter Gelatine so eingelagert, dass ein Virus immer nur das nächstliegende infizieren kann, so dass sich ein Ring bildet, und damit wird gesucht, was sie finden, und dann nimmt man etwas von
what they are looking for. They see where is the action, where something happens, and there they take something, and then they try to purify, and they do this several times. And then they try to, as pure as possible, to get this. And they are successful. They find something, and they make photos there. And they find typical reproducible entities there. And they make photos from them. And it's sure you cannot make a photo with an electron microscope within the mucosa. You have to take the material out, and you have to prepare this. This is always the same. It's a method that has led to so many knowledge about so many very helpful things that I would not leave it just because of your theory.
Let us not get into fighting mode again, but rather agree on those common grounds that we, I think, can agree on. And let us also agree that if you win going your route, I will applaud you. I have absolutely no problem with that, because that is what we all want. We want to finish this, and we want to get the bastards who are responsible.
May I say something, Rainer?
Sure, go ahead.
Yeah, I've been listening on the translation at a delay the whole time, and I think there are a few important points. Just kind of to.
We have Professor Perron waiting, and he's been waiting for quite a while now, unfortunately. So if you for over an hour, so maybe if you could just have a few closing remarks, and then we have to switch to the other guest.
That's all I was planning, and I have had my hand raised for about 20 minutes. Waiting to get into the conversation. But, I want to point out, I know that you all are familiar with Professor Ioannidis, and he published one of the most widely cited articles, which is why more than half of all published research findings are false. And in there, he outlined that how the constraints of the publishing industry, the limitations of peer review and bias, publication bias, funding bias, etc., contribute to false interpretations. And I want to say and emphasize this because I keep being accused of putting forth a theory. I don't have a theory. I'm refuting a theory. In other words, I'm saying the evidence claimed to back up germ theory, specifically with respect to pathologic viruses, simply is lacking. And I haven't heard anyone present any evidence of how the experiments put forth to prove the existence of viruses actually prove viruses. And that is the only really argument that I'm interested in, because of course we agree on everything else, because if there's no virus, then everything else is false. So we agree that everything else is false. We just, are having a difficult time looking at this. And you have to look at this issue as if you're just curious and you don't have any predetermined ideas. Like, I want to know, do viruses cause disease? Let me see. What's the proof that the scientists put forward? Does that prove that viruses cause disease? Yes or no? And that's the only issue that I'm speaking to, not for what is the real or true explanation for an illness, just whether or not viruses cause the illness. And if you look at the evidence put forth, you'll see that they have not met the burden of proof. And I have no self-interest in this. In fact, it's cost me dearly in terms of my relationships, my profession. I was terminated from my position because of saying these things. I was disqualified as an expert witness in court. But nonetheless, why do I keep saying these arguments? Because this is simply the truth. And no one has put forth any logical or scientific argument demonstrating how these experiments prove viruses. So facing no contradictory direct evidence, I feel quite validated that this is actually the closest to the truth that we have about the virus issue. Now, I do not feel that this is a good strategy to use in a legal strategy. In fact, I personally, as a pro se litigant, am suing a couple of different parties who violated my rights individually based on tyrannical COVID policies that are not backed up on science. But I don't need to make this argument, and as you pointed out, it would be extremely controversial. So why is it important, and why is it important to discuss it on this forum? It's because, firstly, this is the actual truth. And secondly, it has severe consequences for our ability to understand and be free from manipulation, as well as for our health. If we keep believing the false narrative and we accept medical interventions based on a false cause of disease, then it will be to our detriment. And this is what has happened with these so-called gene therapy vaccines. So I feel it is the primary issue in the pandemic, and it is argued from first principles of logic, looking at the evidence and simply refuting the primary evidence put forth to prove a claim. Thus, the claim is baseless. And I wish you all a lot of success in whatever legal strategy, because that's obviously your expertise. And I hope that you are successful. But personally, I don't trust the courts to administer justice, and I don't trust them to understand or acknowledge the truth. But I think that we need to use all the strategies at our disposal in order to be able to live a life going forward where we can exert our free will and accept the responsibility for our actions as we're meant to as living men and women on this earth.
I agree wholeheartedly. And they say all roads lead to Rome. Well, probably not quite all roads, but many do. And I think if both of our routes work, all the better for it. And I
Absolutely.
do, and you know what? I personally believe that I'm not convinced that the virus has really been isolated. I don't know whether or not viruses exist or do not exist altogether. But I do think that if it were possible to show that they don't exist, of course that would end the whole discussion. But at this point in time, just like you, I don't trust the courts of law. That's why some of the stuff we're doing is not inside this old corrupt system anymore. For example, this grand jury proceeding we're going to have is outside of the system. We're using an instrument of the old system, but we're doing it outside of the system simply because we do not trust the courts of law. But if you are successful with your argument, then again, I'll applaud you for it, because anything that stops this is right. A doctor that cures is right.
This is just what I wanted to add. You know what? There is a very good book from Heinz von Förster It's a scientist, a very famous scientist who lived in U.S. He comes from Vienna, and he's very clever. And he wrote a book. It's called "Truth is the Invention of a Liar." And it's a very good book because it says that truth is always something, that may be changed later on and be wrong later on because it depends on the views. And he also says objectivity is negotiating, or is that observations can be made without observers. And this objectivity has the idea that observations can be made without observers. There is always this the role of the observer and the interests of the observer, the focus of the observer, the history of the observers, and such things. And this may be very, very different between in cultures, and maybe different from individuals among individuals. And this is why we have this principle of science, which is doubting. And okay, you doubt, but with your doubts, you have to be doubted too, if you want to be in science. And this is what I just insist on.
I think that's a good closing point. I mean, like on a practical level, if viruses didn't exist, what are these people who are busy in like gain of function, activity?
They're designing toxic vaccines.
What are they actually? No, I mean, apart from that, but what are they actually working on? Is it a mirage? I mean, are their jobs just like,
They're
senseless?
working on synthetic genes.
Okay.
That makes sense too. We believe that, or I believe that, what they're doing there, it's either a huge distraction in order to make people believe that whatever they think is out there, a virus maybe, is more dangerous than it really is. I personally believe that this is a relabeled flu, actually. But most people seem to agree that that whatever this bioweapon is consists of two pieces. One is the virus, which they seem to have tried to manipulate, and the other, the real bioweapon, and again, we agree on this one, is of course the vaccines, which are no vaccines whatsoever. But let's leave it at that, and I wish you all the best of luck, both of you, and Tom Cohen as well. With your pursuit of your case, I'm going to try it my way by attacking the PCR test. I think it's going to be a little bit faster, but we'll see. We'll see. I have no problem. Agreeing that you were right.
Okay, thanks so much
Listen,
for this.
what's important is that we're all trying to find the real truth and uncover the lies that come from, the governments, the international organizations, and the psychopaths who are the front people for the whole thing. So thank you for really making that your main goal.
Thank you, guys.
Thank you.
Thanks a lot for coming.
Thank
Have a
you.
great weekend.
Pleasure.
Ciao.
Yeah.
All the best.
Now we